All right. Let's pick up where we left. And I left you with this slide. And this slide signifies the transition from theory to models. So I will tell you now about a model which has a chance of being implemented in a lab and to be actually meaningful in the lab. I don't know why my Zoom is acting up. 1 second. I need to stop it. I need to stop my Zoom. Yes, my Zoom is stopped. Okay. So let me see. Yes. So, right, this part of work was done in collaboration with Veronica Parth and Yulia Friedman. And if the sort of theme for the previous application of the stable marriage problem was pride and prejudice, now I will tell you, oh no, it doesn't want to be stopped. Sorry, guys, just my Zoom is suddenly acting out. It wants something from me. Exit. Okay, now I hope I killed the beast. I killed the beast. So if the sort of the book to follow for the previous application of a stable marriage was Pride and Prejudice, now I will tell you a little bit more about polygamous and more kind of kinky relationships. And by the way, the interesting sideline to the story is that even though we published this paper almost the same time as this paper, originally what I'm going to tell you about, I discovered in collaboration with my colleague Alexei Tkachenko while I was still at Brookhaven National Lab on Long Island, and it was an autocatalytic polymers problem. So it was more of an origin of life problem. Again, if you are interested, talk to me. I will tell you how to connect ecosystems to the origin of life. But then after I started thinking about it, I realized that the model makes perfect sense for a well-known biological systems. So what we will be talking about is communities, microbial communities, which are consuming 2 types of essential nutrients. And I always call it a CNN model, even though it could be CNP or CN iron or PNN. So any pair of 2 essential nutrients essential for life could be substituted for CNN. So what is important is that each species acts as a sort of an AND gate on those nutrients. So no species we know can survive without some sort of metabolite supplying it with carbon and some sort of usually another metabolite supplying it with nitrogen. So the growth of every species could be limited by whichever resource in most scarce supply, and that depends also on the rate at which you can uptake those nutrients in the environment and at the ratio in which this species uses them in its biomass. So I will first connect it to the Akshit talk, which was one of the talks open in the conference, where he taught you some geometrical ways of thinking about the nutrient limitation and growth in microbial communities using so-called Tillman diagrams. Again, Tillman discovered the diagrams in the early 80s, and this is really, really well-written book. So those of you who don't know this book, I strongly recommend. It's well-written, full of illustrations, and again, at least to me, geometrical picture speaks volumes much more than a bunch of equations. So I'm a big fan of Tillman diagrams. The true story is that when I started working on those systems, CNN systems, I was so ignorant about the cutting-edge ecology research that I, together with my colleague Yulia Friedman, actually rediscovered Tillman diagrams, and I was super hyped up. I was saying, now we are writing up a paper, submitting it to high-profile journal, and then I was lucky enough to share it with some of the ecologists. He said, okay, well, bad news, this was discovered by Tillman in the 1980s, and this book has 600 or 800 citations, so that was actually a bit of a downer for me. Okay, so let me actually, could you turn on the light? I will be writing on the blackboard for a little while. Let me set up first with equations, and then I will show you how to interpret those equations using Jesus Christ. he was acquiring citations like crazy. Maybe I'm wrong, but I must have, it must have been 1,000 citations. Anyhow, so let's say we are right now talking in very general terms. We are saying that we have some species which I will call B instead of N because it's bacteria. And I can put 1 over B here, but the growth rate of the bacteria will depend on 2 nutrients, which I will call carbon and nitrogen, C and N. And then as usual, we'll have a dilution rate. So everything is taking place in the chemostat, and the chemostat has a dilution rate. By the way, everything I say would work still the same if species will have their own individual dilution plus death rate. So it doesn't matter, but for simplicity, I'll just keep this delta the same for all species. And then you also need to write down at which rate the nutrients are depleted. So here is one important ingredient. First, let's see how the nutrients are supplied. The nutrients are supplied via a pipe leading to a chemostat, and the concentration of a nutrient, carbon nutrient in this case, in the pipe is C0. But of course, the concentration of this nutrient inside the chemostat is C. If nothing happens, if there is no consumption, then as you see here, the equilibrium state would be that C is equal to C0, but the nutrient is being consumed by a bug, and the way it is being consumed by a bug is B times G of C comma N divided by carbon yield. So the carbon yield is a specific conversion factor between how much carbon is consumed and how much biomass is being generated. So the same can be written for another nutrient, which would be a nitrogen. Again, delta N of 0 minus N minus BG of C comma N divided by another yield, nitrogen yield. Okay. Now let me just now remind you what is the concept of a zero net growth isocline. So what is a zero net growth isocline? When you just look at the growth rate of the species, it could be both positive and negative, or it could be 0. If it is 0, then the species is in equilibrium. And if you think about this line, given the line in CNN space, which separates, which is a solution to this equation, this is exactly what is called zero net growth isocline. I wish I didn't use such big letters to write it. Maybe I'll need to erase it. But for now, let me actually start here by drawing a zero net growth isocline. And let's say this, of course, the shape of this line depends on the exact shape of this function g of c of n. And what Akshit, I believe, told you is what happens when your g of c and n, which, by the way, shouldn't be c and n in this case, but should be 2 carbon sources. So Akshit told you how to think about 2 substitutable carbon resources, for instance. And then the equation would be lambda 1 c1 plus lambda 2 c2. And of course, the zero net growth isocline in this case would be a straight line with the slope given by the ratio of those 2 coefficients. However, what I will be talking about today will not deal with substitutable resources, but will deal with 2 essential resources. And the way to think about how species grow on essential resources is to say that one of them is always in excess. So it's so-called Liebig's growth of the minimum. And the formula for g of c and n in this case would be a mean function of lambda c times c, comma, lambda n times n. So that would be the equation we need to solve. Under what circumstances this mean function is equal to delta? And of course, this is even easier to solve than this linear equation, because we know that this mean function could be either equal to 1 to the left side or to the right side. And which side sets the value of the mean function is what I call a growth limiting nutrient, right? So for instance, when lambda c times c is smaller than lambda n times n, I will say that this microbe is limited by carbon. And what it means is that no matter how much nitrogen I give, as long as this lambda n times n is larger than lambda c times c, the growth rate will not change. And the same situation is when it's limited by nitrogen, then this is the smallest numbers, then I can change the carbon as much as I want, but nothing will change in the growth rate. So the signature of this Liebig growth of minimum is that there are nutrients which I can change the concentration in the environment, and nothing will change. In fact, when people test for which nutrient is growth limiting in the ocean, they literally do exactly this. They sample the ocean water in a tube, and they dump different essential nutrients, phosphorus, nitrogen, and they see which one will result in increase in optical density. If it doesn't result, it means it's not a limiting nutrient. Once you found one nutrient where you will add it to the mixture and suddenly the whole community blooms, that is a rate limiting nutrient for at least one species in the community. So let's say right now we are just studying the two nutrients, and the zero net growth isocline in this case will be this rectangular shape, L-shaped figure. And again, I want to connect it to something which you know. So there will be 2 points here. One which we will call, let's now put names. This will be a carbon, and this will be a nitrogen. And there will be a C star here and N star here for a blue species. So I specifically use the blue color here because the C star and N star depends on the species. So this is exactly what Akshit in his lecture referred to as R star, more generically for R being a resource. And the meaning of this C star, for instance, is that in order for a species to grow, whichever point, this is kind of the way to think about a species. And when you are in this shaded region, there will be an exponential growth of this species. When you are in this unshaded region, there will be an exponential decline in the species. If you are precisely on the zero net growth isocline, the species abundance will not change. And now, if you think about the environment, if you now place the environmental point, if the species finds itself in this environment, the species will grow. As the species will grow, it will consume the resources until eventually it will end up on a zero net growth isocline. And you may kind of think about it this way. So maybe if I start at a particular point in the environment, if I initially, when I imagine the following thought experiment, you prepared your chemostat and you didn't add your species yet. So initially your coordinates of this point are C0 and 0. And this is exactly up to you what they are, because you are deciding how much nutrients to add to your mixture when you are feeding your chemostat. But then you added your species, a little dollop of your species, and started growing. As it started growing, it started consuming the resources. And as you see, the numerator here is the same. The denominators are just constants, which means that it started consuming the resources in a particular ratio. And this ratio is given by YC over YN. So for instance, you may imagine that the species which I added here would kind of grow like this at a straight line until the straight line hits zero net growth isocline, at which point the species will reach an equilibrium, and you will have one species in the environment in the equilibrium. Can somebody tell me in this point, will it be limited by nitrogen or by carbon? Nitrogen, exactly, because it hit the point N star, so you always, and C star, it needs to be, in order for the species to be at this point, it needs to be at the limiting concentration of the limiting nutrient, but for non-limiting nutrient, it has to be larger or equal than C star, okay? And the slope of this line is nothing else but, let me think about it for a moment. So this particular line I have drawn shows that the species consumes a lot of nitrogen and relatively little carbon, because the slope is very steep. Which means that its yield on nitrogen is small, whereas its yield on carbon is large. So the slope of this line would be yc divided by yn, if I am not mistaken. Right. Well, that is because, look, just look at this equation. Whatever is the rate at which it is consuming, in time, of course, there will be a nonlinear process of how it moves along this line. But the amount of carbon and amount of nitrogen it consumes will be proportional to each other, which is, by the way, different than the model which Akshit told you about, the MacArthur model. In MacArthur model, all of those trajectories of the species from the starting point, where you just prepared your chemostat, to the end point will be, I believe, hyperbolas, whereas this one will be a straight line. So you cannot have straight lines everywhere, and this actually is a model which has the most straight line, which is why I like it a lot. Now, let's just now add another species. And another species would be a red species. And I will add it in the following way. So now this is my, my red species, which had its own red N star and its own red C star. Now, let me ask you the following question. By the way, just to connect the notation of N star to whatever I used here, again, remember that I have those two coefficients, lambda C and lambda N. So N star is nothing else but delta divided by lambda N, whereas C star is nothing else but delta divided by lambda C. And of course, you know, I'm not making my drawing too complicated, but you have to remember that the blue value of lambda C is not the same as the red value of lambda C. Could you tell me which of those two species is more competitive, you know, will outcompete the other one if the nitrogen is a limiting resource? Which one is sort of
red?
Red, yeah, exactly, because the red will drive the concentration of nitrogen to the lower point. Another way of thinking about it is that if I imagine myself being somewhere on this red line, all of the points on this red line are below the blue line, which means that if I'm on the red line, on this branch of the red line, I am in the region of the exponential decline of the blue species, which means that the blue species will die if I'm somewhere here. But now I'm actually at the opposite point. I arrived at this point, and according to this point, the red species would grow because this blue point is in the growth region of the red line, which means that at this point, the equilibrium will start to move from the blue point towards what it will be moving towards. We need to determine a little bit carefully. So for this, I will kind of draw yet another line. I will draw a line. What if I added the red species first instead of the blue species? So then I assume here that the slope for the red species would be different from the slope of the blue species. By this, I'm inherently assuming that the two species consume carbon and nitrogen in different ratios. If they were consuming in the same ratio, the model will be degenerate. So for the model to be really interesting and rich, you need to have the species with different stoichiometries of essential nutrients. So here, if I just added the red species first, I will arrive at this point at the zero net growth isoocline, which again shows that if I will add the blue species later, if my environment was in this point, this red point is in the growth region of the red species, which means that the blue species will now start growing, which kind of are two experiments I can actually do. And they will start growing until, you know, those are two different experiments, until they will arrive at this intersection point. This intersection point is in some sense unique because this is the only point in the entire plane where two species can coexist. And in this case, this would be indeed the coexistence point of two species. So it doesn't really matter in this case in what order I will add the species. If I will add a little bit of each one of them, so one way or the other, the community will arrive at this coexistence point. So again, I might have kind of started mixing the species right away when I just was preparing my first inoculum to put in the chemostat. If I put a lot of the red species and very little of the blue species in my inoculum, my trajectory by which I arrive at this point will be something like this. it's no longer line because the species, add up and they have the different prefactors. So as those prefactors change, the slope of the line also changes. If I added a lot of the blue species and very little of the red species, my trajectory would be something like this. If I very carefully calibrated the ratio, I might actually do even something which looks more like a straight line. Okay, so this is how you reach an equilibrium in this community. But now I want to show you that this fact that you will arrive at this coexistence points is not a God-given fact. Yes.
So to the left of the red C star line and the red C star line, yeah, and below the blue N star line, those are single species areas, right?
Well, let's put it this way. First of all, right now, I'm not asking the question, which I will be asking in a moment, so I will be able to answer your question in a moment. I'm not asking the question of what will happen if I will start changing C0 and N0, which is a very good point. What if I start messing around with the ratio of nutrients in the environment in my chemostat, where I will arrive? Right now, I'm starting here, and I'm just saying that my different black lines are different ratios of the species in my inoculum, in my dollop of, and they will not cross those lines.
Right.
However, a very good question now is what will happen if I will move this blue point somewhere way left or somewhere way right? And you're right that I'm not guaranteed to arrive at this coexistence point. That is a very good question. But before we address this question, let me show you that it's not even a given fact that I will arrive at the black point at all. I want to now change my species a little bit. Namely, I will not change their zero net growth isoclines. So I will not change their own growth, but I will change their stoichiometry. So what if the blue species actually has a strong yield on nitrogen and weak yield on carbon? So what if the blue species will have this slope? Then again, if I just added the blue species alone, it will kind of follow this line until it will arrive at this fixed point here. And when it arrives at this fixed point, notice that if I will add the red species later, red species will not grow, right? Because this cross here, X marks the spot, is in the no growth regime for the red species. And vice versa, I can have the red species, which will arrive right here at this X, which will be below the zero net growth isocline of the blue species, which means in this case, for the same growth rate, for the same G of C and N for both species, I will have actually a bistable situation. I will have either the red species wins or the blue species wins. You may say, what if I carefully prepared my environment that I am at this point? Can somebody tell me what will happen if I'll just carefully put everything at this point? It's unstable fixed point, exactly. So depending on just a fluke, it will either flow to the red or to the green or to the blue, and that is all determined by those two lines. And now I'm happy. Now let's assume that I'm going back to the. Actually, why don't I just now use this for an example, right? So now I can, actually, let me now go back to my previous example where I had a coexistence. When I had a coexistence, I had something like this, and like this. Now I'm raising my old axis, and I have those 2 points. Those are the 2 single species fixed points. This is what happens if in this environment I only had 1 species Now, let's just say the following. Let me take this coexistence point and draw a blue line parallel to this dashed line, blue dashed line, and I will draw a red line parallel to the red dashed line. Now it kind of divides my entire CNN plane into several regions, right? So for instance, there is this region which I will shade in black. Now, what happens to the species if my environmental starting point, C0, N0, is within the shaded region? Can somebody tell? It will flow here. Now, how about here? If my initial point will be here. Who will survive? Red. And here, only blue will survive, okay? And by the way, if I will start anywhere here, both species will die. This is a death zone, so that is when my dilution rate of a chemostat is too large, so the species will be washed away, and if I start here, nothing will happen, my species will be washed away. So this is kind of the gist of the zero net growth isocyclines and how to work with them, and the take-home messages here are that you need to know two things. If you want to know the state of the environment, you just need to know, you just need to know the zero net growth isocyclines, and you can determine this intersection point, if it exists, if they intersect at all, in order to see what would be the coexistence state. But then you need to know the yields in order to know whether this coexistence point is stable or not. So dynamical stability is not guaranteed, and it's hidden in some inequality on the ratios of carbon and nitrogen supply. I want to make one more point using this zero net growth isocycline, which I will use later in my talk. And this is a point about invadable and uninvadable states, which is also quite useful, and I believe it has been discussed in the concept of, in a context of Lotka-Valterra, but has not been really discussed in the context of consumer resource models much. So let me just, I need a bigger plot here. It's a mess, but it's okay. So let me start with a bigger CNN plane. And fortunately, I have three colors, so I can work with three species. So let me say that this is my C, this is my N, and I might have my friends, which I already forgot in what order they were, like this. And let's say I will have the red one like this. And let me first show you a green one like this, okay? Can you tell me, based on this figure, if those species can, you know, if one of those species would be present if I kind of keep adding them all together, would one of the species be completely obliterated in this picture or not? Yeah, red will be a goner simply because it is worse than the green one on both carbon and nitrogen. So red is a goner, so we'll not keep it. You may imagine that you added, I don't know, 100 species by randomly selecting those lambda C, lambda N. Some of them will just suck, but some of them will not, and there will be, sooner or later, there will be another one who will be added to this community. Okay, red one. Now, you actually have something interesting. So I may imagine my community, if I first just added the blue and a red one, the community will end up at this point. Can you tell me, and then I'm mixing in to this mix, I'm mixing in the green one. What will happen? The green will grow, right? Because the way to look at it is this point is within the L-shaped form of the green species, meaning the green will start invading. So for sure the green will invade. Who will be displaced? It's not at all obvious, but green will definitely enter the mix. And since there cannot be more than two species in the mix, it will flow either to this point or to that point, right? But now, which by the way means that this point is invadable. In this collection of three species, this point is an invadable state because the green species will invade. On the other hand, this point and this point, those pairwise coexistence between blue and green red are uninvadable. And we are naturally more interested in uninvadable points than invadable ones, because if you think that all species have some chance of being introduced into the ecosystem, invadable points are inherently unstable, because the moment you introduce even a dollop of the green species to an invadable point, it will outcompete either blue or red. So throughout my talk, I will tell you how to count the number of uninvadable states here in systems like this, and also how to predict sort of what kind of transition will be between them, whether it will be a regime shift between, or it will be just a peaceful coexistence, which could happen. If you are kind of in this situation where the coexistence happens, where you, for instance, now let me get rid of the invadable and uninvadable mass here, and the green species also. So if I just go back to my original plot of the red and green, and if I now, I will try not to mix it up. If I now plot the blue line here and the red line here, as we discussed, there will be this pizza pie shaped region where if my environment, if my C0, N0 is inside this slice of a pizza pie, then it will lead to the coexistence. If it's outside, it will lead to the either red species surviving up here or the blue species surviving. Now imagine I do the following experiment. I select some line along which I will gradually vary the C0 to N0 ratio. So my environment will vary from very carbon rich to very nitrogen rich here. So what will happen when it crosses this point? So first I have only blue species, right? What will happen at this point? Suddenly red species will appear. When you say suddenly, would it appear in a large quantity? No. And that is very important. So there is now I'm switching to the physics jargon, there will be a second order phase transition on this line, right? So there will be a continuity there will be an order parameter when you cross this line, namely the concentration of the red species, but it will be a smooth transition. And then those two species will exist. And then at this point, the blue species will also gradually go down until it hits zero. So there will be two sort of second phase transition lines. But if on the other hand, it was reverse, so if I go back to my bistability, then I will have a first order phase transition. If I will go like this and like this, then suddenly the picture will be very different if I change my nutrients along the same line and I follow the concentration of one of the species. It will be something like this. Something like this. So for instance, the blue species, let me see if I did it right. Did I did it right? Yeah, I did it right. So the point is that if I'm here, I have only blue species, and the blue species will persist until it suddenly, very abruptly disappear here, drop to zero from a precipice. And then when I go back, I need to go back down here until the red species, the blue species will reappear. Yes.
I have the impression that this is the opposite model. If you start at the bottom, if you follow the red line from the left, and if the red was known, the bottom right part, lead to a point where two can grow.
You may be right. You may be right. You may be right. Again, I'm messed up with colors, and thank you for pointing this out because it wasn't at all obvious to me. So indeed, this is the domain of the red species because if let me just think about it for a moment. So when I'm down here, my nitrogen concentration is low. That means, yeah, it will be a red species which will persist up to the point I get here. Then it will suddenly drop, and the blue species will drop at this point. Okay, now we are on the same page, and that's all I wanted to illustrate in terms of this generic Tillman diagram. Now I will show you how to connect it to what I told you before, namely how to actually enumerate the number and identity of those stable states in a more complicated setting. Because of course, this is idealized pictures that you only have carbon and one carbon and one nitrogen. So let's see where I will go with it. I had a sort of, let me just scroll through this. Okay. So now the model which we actually worked on for, as far as I know, for the first time, is a model in which you still have only two essential resources, C and N. However, each of those resources is represented by multiple different metabolites. Imagine you have K sugars or other carbon sources, maybe also organic acids or something, and M different nitrogen sources, NH4, NO3, and so on. So you have all kinds of different carbon and nitrogen sources mixed in the environment in different ratios. So the ratio is, again, something we control. I will assume no cross-feeding. That's one approximation which I do here, because that's the only one which allows me to do a reasonably simple model. And that's not a huge crime. The other kind of approximation is that I will only consider specialist species. So each of my species, each of my microbes in the environment is capable of consuming exactly one carbon and exactly one nitrogen, right? So they are all specialists, but there will be no species which only consume carbon, because both carbon and nitrogen are needed for life, but all of them will consume exactly one carbon and exactly one nitrogen. So, which means that in principle, there are K times M possible species types, right? You have K types of carbon mixed up together with M types of nitrogens. But now if you think about the competitive exclusion principle in this case, I have sort of in my G of C and N, I will have K C variables and M N variables. So altogether, I will have K plus M variables. So by definition, unless some miracle happens, I cannot have more than K plus M species surviving. So out of this K times M, I only have K plus M survivors. So most of my species will be doomed in any given environment. Yes?
Once more, why there are only K plus M species surviving now?
Very good, very good. I will just go through it it is a very basic fact, but I want you to understand it because it's really, really fundamental fact. So when I'm saying the species surviving, I need to solve the following set of equations. So now I have the species labeled with alpha, and each species has its own growth curve, and the growth curve depends on N1, N2, and so on, and M. By the way, I'm writing it in the most general term, which may include also non-specialist species. All of my species will only depend on one carbon and one nitrogen, but that doesn't matter. The competitive exclusion still applies. I need to solve this equation because remember, zero net growth isocline is defined by this condition. So, and again, that's what I'm saying, that I don't allow for any miracles. I cannot have more equations satisfied than I have variables here. So I have K plus M variables, but I can have more than K times M species. I can, for each combination of carbon and nitrogen, I can different lambda C and lambda N. So I can have gazillion species, but in any given environment, no more than K plus M of them can survive, because in order for them all to survive, they need to be on zero net growth isoclines. And I have K plus M variables, so the maximum number of equations I can solve is K plus M. This is very, very interesting. By the way, I will not go into it today, but if you're curious, ask me over a coffee break, what are the consequences of this for the origin of life? And I will give you a teaser. That means that no matter what kind of alphabet of the letters you are using in your DNA, RNA, or maybe some proto-RNA had more letters than 4, in the end, you always end up with a binary alphabet. And that is all due to the competitive exclusion principle, because you are kind of out of Z square possible combination of letters, you select 2 Z letters, but I will not go anymore because that will distract us. Okay, so now the final thing which I want to introduce as a definition before I move forward is that I will be talking about a state, and how the way I define a state is I have a pool of species. So a state, yes?
Sorry. So this is K plus M is true only assuming that every species has a distinct carbon and a distinct nitrogen source that it depends on.
No. That is why I wrote I'm saying that whatever the growth equation. So each species, you have an environment. In the environment, you have K carbon molecules at some metabolites at some concentration, M nitrogen molecules. And each species has some growth rate, which is a function of those concentrations. I don't allow for anything else. I don't allow for pH. I don't allow for temperature. Just as long as the growth rate is only determined by concentrations of the nutrients, of the resources in the environment, all I need to do is to count how many nutrients, resources I have in the environment. And by definition, I cannot solve molecular equations and I have variables, right? In fact, the very fact that I am dealing with those specialist species doesn't help me in any way. It makes this equation maybe a little bit easier to solve, but it doesn't change the fact that I cannot generally solve more equations than I have variables for. Okay, now the last thing which I wanted to tell you about is that my definition of a community state in this model is slightly different than what, for instance, Lotka-Volterra people will do. Lotka-Volterra people will say a state is, if I have a S species, I can potentially have 2 to the power S states because each species may either be present or absent. This includes the trivial state where all S species are absent. My definition of the state is I want to know not only which species are present or absent, but also which nutrient, which resource, carbon or nitrogen, they are limited by. Which means that in my model, the number of states is potentially 3 to the power S, not 2 to the power S. So the species can be absent, or it can be present limited by carbon, it can be present limited by nitrogen. If I change my variables and everything remains the same, but one species changes its nutrient limitation, which can happen, for instance, if only one species is present, I will say that it changed the state. Okay. Now, this is like a pictorial way how I represent it. It's a busy plot, but let me walk you through it. And maybe I can now turn off because for a little while I will not be using the blackboard, the whiteboard. So let's see. This is my definition of which nutrient is limited. By the way, the microbes are here. I don't know if you see those little wheels. Those are the microbial tails, flagella. Those are my 2 carbons, 2 nitrogens. Those are supply rates, which are concentration of nutrient times delta times dilution rate. And when I'm looking at a state, I'm signifying which is the limiting nutrient. And I do it by drawing this oval shape here. So which means that in this particular state, this species 1, 2, meaning that this is a species consuming a carbon of type 1 and a nitrogen of type 2, is limited by its carbon source. This species 2, 2 is also limited by a carbon source, and this one is limited by a nitrogen source. Now, let me just first illustrate this concept of states using a toy example, which we described in the paper. In fact, this is, even though the example is as simple as it can be, 2 carbons and 2 nitrogens and 4 species, because 2 times 2 is 4. It already has quite a rich variety of possible states. It has as many as 7 states. If you only count uninvadable states. Remember this distinction between invadable and uninvadable. The one intersection between 2 things was invadable, but the 2 others were uninvadable in the example I have shown you before. So those are the 7 uninvadable states, and those are the states I really care about. Whereas invadable states, they may be kind of transient, but if I keep adding species all the time, they will sooner or later disappear. But just for the record, this whole thing also has 27 invadable states. And again, you know, as you see, states have different diversity or richness. Those 2 states have only 2 species. There are 4 3-species states and 1 4-species states, which for the selection, you know, a priori, this is the information I could have told you without knowing many details about the model. All I needed to know, just like in a stable marriage, are preferential sort of rates of the order in which competitiveness of different species on different sources. So all I need to know is the rank order of this competitiveness. So let's say if a species 1, 2 outcompetes the species 1, 1 when they are competing for a carbon, I will put the 1, 2 to be rank 1 and 1, 1 to be rank 2 or something like this. So this whole information I could tell you, I could predict to you, just like in a stable marriage, only based on the relative ranks of competitiveness, which is a great boon for us because we all know that it's very difficult to measure things in a chemostat. If you approach an experimentalist and tell tell your experimentalist friends that I want you to measure those lambdas, they will run away from you like you are like you have some infection, because takes a lot of work to measure those lambdas. However, these competitive exclusion experiments are relatively easy to perform, and this rank ordering is not that hard to determine, even in the chemostat. So I can predict those states that I will predict that those 7 states will be uninvadable just based on those rank orders. What I cannot predict without additional information is in which region of the fluxes each of the states will be realized. Because remember that there will be nowhere a situation where all 7 states will be simultaneously possible. If you mix your carbons and nitrogens in a particular ratio in your chemostat, usually the most common outcome is that only 1 state will be possible. Sometimes you will have 2 states, just like we had in the bistability example, where in the center of this pizza pie during the bistability region, I could end up in a state 1 or in a state 2. And again, the more nutrients you have, the more possibilities you have. You can have a tri-stability, you have a quadra-stability, and so on. And the other thing, so I will not be able to predict it unless I know yields in addition to the, unless I know the yields in addition to the ranks of nutrients. And finally, the other thing which I cannot predict is which of them will be dynamically stable, which of them will not be dynamically stable. And that you saw the example on my toy example, because I had the same growth curves and the same intersection of the growth curves, which in one case was a stable community state, which corresponded to the 2 species coexisting together. In another case, it was dynamically unstable. So in this particular example, which I used in the paper, the state number 7, which is the richest state with 4 species, turns out to be, unfortunately, it turns out to be dynamically unstable. So altogether, I'm left with 6 dynamically stable states. And if I now start changing my ratio of my 4 nutrients, I can have all kinds of transitions between our 6 states. I can have the transitions which will be first order or regime shifts. And between some states, I will have the transitions which are second order, where you will just gradually, one species will disappear and replaced by another species. So this is kind of phenomenology of this model. Now I will kind of connect it to what I was talking about before. So in the paper, we developed an algorithm. And again, we didn't embark on this project looking for a stable marriage solution. We embarked because we liked the problem. And then against all odds, 2 times in 1 year, I found 2 problems which mapped on a stable marriage problem. So talk about the karma or anything. So it was clearly my karma to discover stable marriage everywhere on this particular year around 2018, 2019. So as I already told you that all you need to know is relative ranks of those lambdas, right? Remember that I told you that for 2 nutrients, I have, say, species alpha will have the growth rate given by lambda alpha C times C whatever I, which it uses, comma lambda alpha N for whatever N J it uses. That will be how the growth functions. And this will be, by the way, this will be actually mean function. This will be G alpha would be given by the mean function. And I already told you that Ci star will be given by delta over lambda Ci alpha star by lambda alpha, and J star will be given by delta over lambda alpha n, right? So when I'm saying that all I need to know is the relative ranks, I mean, out of all the species which are using a given carbon source, each one of them will have their own lambda. Whichever of them will have the largest lambda will have the smallest C star, and therefore will outcompete others. So my rank, I'm kind of more comfortable thinking about it in terms of lambdas than in terms of C stars. So I will keep thinking in terms of what is the largest lambda, and the largest lambda means the most competitive species, right? So I would say that in this jargon, the most competitive species, a species with rank 1 for a given nutrient, is the one which has the largest lambda alpha, but I only include the ones which are capable of using this nutrient, so say C alpha. And the weakest competitor will be the one which will have the smallest lambda. So in order to determine those uninvadable states, I need to actually use a special variant of the medical school, which was in the original paper, in 1962 paper, and it's called the hospital resident algorithm, which is algorithm to determine those hospital residence assignments is exactly the same as a stable marriage we played. The difference here is that whereas the marriages were one-on-one, here one side is actually the hospitals are, you may call it the polygamous, and this one is monogamous, right? When you're applying for a job, you end up getting one job. I hope you're not cheating and getting money from two places. But when your university is hiring, routinely they have more than one position. The same story with hospitals. When it is the season to hire medical residents, each hospital announces how many openings it has for residency. And some hospitals may sit out this round. This hospital in particular has 0 openings. This one has 1 opening, and this one has 2 openings. By the way, in this case, it doesn't need to be that the number of hospitals matches the number of applicant students, as well as the number of openings doesn't need to match the number of candidates, even though it will be just like in a marriage problem when there is a mismatch between the sum of the numbers on the left and the sum of the numbers on the right, that will have a severe consequences. So in my problem, fortunately, the sum of the numbers on the left will be equal to the number of openings is equal to the number of candidates. So everybody will find their job, you know, in this round. But other than this, if you are doing, for instance, applicant propose an algorithm, they will just apply to the hospital of their top choice. All the hospitals do is they receive applicants, and if the new applicant is better than their worst candidate, they kick out the worst candidate and and get the new one. And it converges according to the same logic and the same rules as a stable marriage, which is one-on-one stable marriage. So now I will show you, and that is a little bit of a heavy lifting. Yes. Oh, no. Also, hospitals will rank candidates completely differently. Again, in reality, maybe they will, if they use the same matrix, maybe they use the GPA or whatever other stupid things the hospitals use. But just imagine that those are hospitals which also take into account where you're living and what you wrote in your essay. So the hospitals will create their own ranking, which doesn't have to be correlated at all. And so will applicants. The only difference between the sort of the stable marriage and a hospital or college admission problems, as Goyal and Shapley formulated it, is that in the marriages, it's always one-on-one. In hospitals, one side is polygamous and one side is monogamous.
Correlated lists can be analogous to
well, uncorrelated, yeah, uncorrelated in some sense, it just means that in fact, in my model, there will be, I haven't yet described how to map, so it will be very soon, but I will answer your question. So uncorrelated, this means that there is absolutely no universal kind of competitiveness of a species, but the species, remember, in my problem, well, I'm kind of jumping ahead of myself. Maybe I'll just answer this question and I'll move to the next slide. Yes.
Yeah. So maybe you're going to talk about this very soon, but you said that the sum of the numbers on the left and the right side are the same.
Yes.
In this case, so just to drag the metaphor into the resources a little bit more, does that mean that there are no resources left unconsumed? Everything is consumed by
no, it doesn't mean, and we will see it in a moment. So this is actually, you know, for now, just take it as a hospital problem. The hospital problem here, for instance, this hospital declared that it will have a no vacancy. It doesn't mean that this resource will be unconsumed, but it gives it a better chance to be unconsumed. So let's talk about it in a moment. So let's say this is now how this hospital ranking problem maps to my, so my goal right now is I have certain number of carbons, certain number of nitrogens. Number of carbons doesn't have to match the number of nitrogens. Here I have 6 and 6, but it absolutely doesn't matter. I can have 3 5s, 3 10, 3 100, whatever. So now I have many microbes. Again, in the simplest case, I will have as many microbes as I have a pair of resources, but my problem is generalizable to the case where the same pair of resources might have multiple microbes. So this doesn't matter. For now, just for simplicity, I will stick to the case where for every pair of resources, there is exactly one microbe which consumes this pair, but it's not important. And my job is to find all uninvadable stable states, which might be actually stable is the wrong way to say. They might turn out to be dynamically unstable, steady states. I know that my job is to find all the states. If I carefully prepare my ecosystem in, it will stay there for forever. But if there are some fluctuations, it may be kicked out from the state if it's dynamically unstable. And the first thing which I wanted you to notice, actually, this is one little point which I forgot to make. If you look at all of those states, all uninvadable states which I enumerated for this simple model, you may have noticed that in all of those states, either nitrogen side or carbon side, or sometimes both sides, like in this state, every single nutrient was limited to one microbe. So it turns out to be not a coincidence. This is a deep rule here, and I will explain what this rule is, what kind of rule, what causes this rule. What is causing this rule is that imagine that in my system, now I have many more carbons and many more nitrogens. Imagine that I have one carbon source and one nitrogen source, which is not limiting to any of the microbes. If the source is limiting to some microbe, I know exactly the concentration of this source, and it is low. It is given by this delta divided by lambda. If the source is not limiting to any microbe, generally speaking, its concentration is high. It's something of the order of the flux rate with which I provide nutrients to the environment. If I had a state in which there was at least one carbon which is not limiting to anyone, and at least one nitrogen which is not limiting to anyone, the species which consumes those two resources would grow exponentially if I introduce it to the system, right? Is it clear? Because they are all in high concentration, they are not limiting to anything. Therefore, if I introduce a species, this growth rate will be way larger than delta. They will exponentially grow, and that means that the state will be actually invadable. So in uninvadable states, it's always either left side or the right side is limiting to at least to exactly one microbe. Because the other thing which you may notice is that the limiting side never has more than one microbe limited by it. For instance, here, this microbe is limited by N2. This microbe is limited by N1, but I don't have yet another microbe which also is limited by N2. And that is kind of easy to understand, because if I have two microbes which are limited by the same nutrient, only one of them will survive, because one of them will always have the R star lower than the other. Therefore, one of them will always outcompete the other. So the bottom line is, in all of those states, the one side has limiting nutrients and one microbe sort of connected to a limiting nutrient. The other side, it's free for all, because there is no principle against by which I cannot have the same carbon being used by more than one species, as long as it is being used in a non-limiting fashion. This is a subtle point, and I realize that many of you probably will be lost at this point, but okay, for now, let's just trust me. And for now, I will just show you the following two things. Actually, stable, unstable, I don't know. I'm saying that it will be a steady state. I mean, what I'm right now enumerating are steady states which are also uninvadable, meaning that steady state only means that if I carefully prepare my ecosystem in this state, it will remain there forever. But if it's dynamically unstable, it can be kicked out by a smallest perturbation. Uninvadable means that I can try every single one, or in any combination, the microbes which are not present in a state. And if I add them to the state, they will be outcompeted. Those are the states I'm after. Yes.
So in the first case where there's at least one carbon source and one nitrogen source, which is not consumed by any of, which is not limiting for any of the existing microbes, the introduction of a new microbe which consumes these two will only be invadable if the R star of those two is lower than everything else that's in the system already. Right?
Well, eventually it will invade this microbe, which you know, this thought experiment which we carried out, introduce this microbe which consumes exactly those two non-limiting resources, will mean that it will invade the state and will be limited by either one or the other. But since it is already invading, it already makes my state invadable. That's why, since my job right now is to enumerate and show you all the uninvadable states, this is not possible in an uninvadable state.
Okay, so basically invadable does not mean that there's extinction associated with it. It's just that there's
well, yeah, invadable, by the way, yeah, absolutely. Invadable can mean, a good example of an invadable state is I have one microbe only. I add one microbe, it happily lives there, but it's still invadable, right? The state has changed.
Right.
That's good. Thank you for clarification, because indeed, in my illustration here, I used uninvadable state where adding a microbe led to extinction. That was kind of maximal diversity uninvadable state, but sorry, invadable state. But invadable states come in known flavors, including the simplest invadable state will be empty state, where the ecosystem is empty and it's invadable by any species. Okay, now. So it turned out, and I can see that it's already probably, given the fact that I have half an hour left, I will lose all of you by giving you all the details. So for now, I want you to trust me. And again, if you're really interested, talk to me after the lecture. So the way to determine all uninvadable state consists of the following two steps. Well, actually, there is a step 0. I need to decide which side will be limiting and which side will be non-limiting. So in this particular case, nitrogen side is limiting, and that means that every nitrogen has exactly one species limited by this resource. Those species now are depicted as arrows, right? So for instance, N2 is limiting to the species consuming N2 and C2. N1 is limiting the growth of the species consuming N1 and C2. So the out degree of those nodes is 1. The in degree of carbon sources can be anything, right? For instance, in this example, the in degree of this carbon source 2 is 4. The in degree of carbon source 5 is 1. And other carbon sources are not used by any of those species, right? So it turns out that in order for me to find uninvadable states in this problem, which have this kind of pattern of nitrogen limited. Notice that I'm only showing you right now nitrogen limited species, and that is why it is step 1. All I need to do is I first need to decide, oh, by the way, I forgot C1. C1 is also consumed by one species. I need to come up with a combination, any possible combination of numbers like 1, 4, 0, 0, 1, 0, as long as it adds up to the total number of nitrogen sources. So for every configuration, I will have at least one uninvadable state, right? So imagine that I decided first on those numbers. I decided that I will be looking for uninvadable state where this is 1, this is 4, this is 0, this is 0, this is 1, this is 0. And then I need to run the stable marriage problem, in which those guys are college applicants, and those are, or medical doctor graduates, and those are hospitals. And this is the number of vacancy a hospital has. So I just need to run this whole thing and find all stable states, not just the applicant proposing stable state, all stable states. For each stable state, there will be exactly one uninvadable state in my problem. Yeah, I cannot change this fact. Outgoing degrees is always 1. For each nitrogen, I will have exactly one species limited by it. But those numbers are up to me, and since they are they are actually 2n minus 1 choose n of those possible ways of assigning those numbers. There is a lot of possible uninvadable states, exponentially many. But for each configuration, in order to find them, in order to find which species actually can survive, I really need to run my whole college application, which is still computationally much easier than looking for a needle in a haystack. If I really wanted to find it without the knowledge about the college application problem, I will have to investigate each one of the 3 to the power s possible states. But this simplifies it much. It's still, it's still a formidable problem, but it simplifies it a lot. Okay, but now Why it is an uninvadable state? Well, because if I'm trying to add a microbe, this green microbe, by the way, so far, what I want to show first is that it cannot be invaded by a nitrogen-limited species. So imagine I'm trying to add one more arrow here, this green arrow, which is still limited by nitrogen. Obviously, this is impossible to have more than one species limited by the same resource. So this one, if I successfully add it, will outcompete this one. But I'm just asking right now, would this green species grow or not? In order for this species to grow, its nitrogen concentration needs to be above its N star for this species, and its carbon concentration needs to be above the C star for this. Both conditions need to be satisfied simultaneously. If you now think about it, it's exactly the condition for this break-in pair in a stable marriage problem, because in a stable marriage problem, we were asking, can two kind of one man and one woman who are not currently married, can they, escape with each other? And they will escape with each other as long as each one prefers the other one to its wife or to its husband. And in the hospital problem, it's slightly modified, meaning that in order for this green species to be accepted by this carbon source, you need to have R star smaller than at least one of its current residents. Okay, so okay, now this is only half of the problem. I need to go to step 2. Yes. Well, that is not possible because this particular state has been constructed by a stable marriage. This is stable marriage state, which I constructed by Goyal-Shapley algorithm and then possibly breakups and divorces. And the stable marriage problem exactly means that there is not a single unmarried couple, which in this case N5 and C2, which can escape with each other. This is just the same condition for stability as for invasibility here. Because in order for a green species to grow, it needs to be simultaneously better than this species and better than at least one of those 4 species, right? So it is exactly the same condition. So far, I only demonstrated that this state cannot be invaded by a nitrogen-limited species. But I can easily add, for instance, to this C4, I can easily add a carbon-limited species as long as it's better than the nitrogen resident. For this, there is a step 2 in the algorithm, which means that it turned out, and we figured it out with my collaborator, Julia Friedman, that there is a unique way to add a unique configuration of the carbon-limited species, like the ones shown here, which will prevent the state from being invadable by carbon-limited species as well. Again, this will kill you if I will explain you, so I will spare your brain. Yes?
Staying with the nitrogen-limited case, how constrained is the definitions of which carbon sources and nitrogen sources?
That is actually a good question. We haven't thought about it, but I believe that if some of the carbon-nitrogen combinations are impossible for whatever biological reasons, you need to work with a stable marriage in which these guys are impossible. But this is actually not a big deal. In any case, it just means that the rankings will be modified. Whenever I'm ranking, if something is just not present among my list of species, this particular space will be missing. That's not a big deal. I can also have multiple species. I already told that I can have more than one species using the same pair of resources. Just means my ranking list will be longer. I have to be a little bit more inventive in my notation, but that's mathematically, it's not a deal breaker.
So the ranking can be
yeah, missing pairs or extra pairs, which I always say that, 2 people can be married in more than one way, which interpreted whichever way you want. Okay. Now, so there is a unique way how to kind of lock the species. So I already made a state which is uninvadable by nitrogen-limited species, and now I can need to carefully select carbon species. In a nutshell, the carbon species, the ones which will set up the concentration of carbon sources, I need to select them in such a way that all the blue arrows which I selected are possible, meaning that this rank of this blue arrow on this carbon source needs to be better than the rank of the red link here. as I kind of jokingly explain this model, I'm saying that this is a model with wives and husbands and lovers, and it's allowed to have as many lovers as you want, as long as they are better than your spouse. So that is exactly the rule, that this is sort of a lover, and it has to be better than a spouse. Okay, I will leave you with that. Now, okay, fine. So we have the unique mapping, one-to-one mapping of uninvadable states in a hospital problem, with the extra kind of caveat that I need to go over all possible configurations of those numbers, of which there is 2n minus 1 choose n. And for each one of them, I need to solve a stable marriage problem. So it's still a formidable computational task, but much better than evaluating it just from scratch. And well, first of all, it's an example. I really drew those arrows completely ad hoc. But what is also important, and also this is quite rare case, because if you will see that all of the carbons have at least one limiting limit, at least one, well, actually precisely one, because carbons also cannot limit more than one. But it is possible that for some of the carbons, they will not be limited to any species. That's why in this particular case, the diversity of the ecosystem is 12. 12 species, 6 species limited by nitrogen, 6 species limited by carbon, but generally speaking, it can be below 12. So it is not more complicated. In fact, if anything, it's easier. Once I know the blue species, I can find the red species in a simple algorithm. I basically need to find the first surviving species, which is more competitive than on incoming species which are using this. So again, the logic is that, well, for instance, look at this carbon 2. It is used by 1, 2, 3, 4 species in a non-limiting fashion, but it also has one species, this red species, which is limited by it. The red species is the one which sets up the C star. The 4 blue species are just leaving off the excesses. So they are consuming this carbon. So they need to be encountered when I'm writing the mass action or whatever. But they can be, completely ignored if they have no influence on the concentration of this resource. Concentration of every resource is set by unique arrow emanating from this resource. If there is no arrow, this resource is present in high abundance, much higher than, comparable to what I have in the medium. Maybe a little bit reduced by the species consuming it in a non-limiting fashion, but definitely not at the, usual chemostat setup is delta is fairly small. And as a result, the concentrations are all such that they are of order of Monod constant of the species living there. That's why chemostats is a completely different game than the serial dilution systems, which I will be talking about tomorrow. Yes, any questions? Yes.
This is a dynamic question that it's not, but as you're saying, like just C2 is simply by the fact that its in degree is higher. Even though it's not limited by that many species, does the fact that it has a higher in degree also not make it a more limiting resource on the entire, on the community level? Does that even make sense?
Well, that's actually funny, common sense tells you yes.
Yeah.
And this is, by the way, it means that if I am just randomly selecting, remember, I didn't talk at all about how to realize this state. There is a unique recipe for how to realize this state. And when I'm saying realize, I need to select my fluxes, my concentrations of carbon and nitrogen sources in my pipes leading to chemostat in order for this state to become a reality. So this nutrient is used a lot. It's used by 5 microns, 4 non-limiting way, 1 limiting way. I need to supply a hell of a lot of it in order for it to be actually feasible. But this is a feasibility question. And the feasibility questions are as easy in this model as they are in a Lotka-Valterra or in my Carcer model. It's not easy to predict for a given set of fluxes which state will be realized, but it's very easy to solve inverse problem. If I want to realize, so if I am really interested in realizing this state, moreover, I'm interested in realizing this state with specific abundances of microbes, I can just plug in abundances of all the microbes which are surviving in this state, all of this red and blue arrows, multiply them by some matrix and get a voila, here are the flux, the concentrations of nutrients in my pipe in order to realize this state. That will make it feasible, and that will answer your question about how come that this guy is used by so many and yet not limited. I may have even, it is possible that there would not be a single red arrow to C2. It might not have been limited to anything, but used by many. So it is possible. And that is why this limitation is a subtle game which involves all. Okay, I need to rush through the rest of my slides, but I went through. Okay, so this slide is easier. This is actually the numerical, the red points is what I was talking about. The black diamonds is the total number of states, including invadable ones. I can also enumerate them, but that's kind of much more boring because they are much less relevant. We can predict them from the stable marriage model. In fact, I can do either naive lower bound estimate by saying that everything has exactly one stable state, or I can do a saddle point integration, which takes into account the proper stable marriage calculation. I did both of those calculations, and both predict the numerical results fairly well. Now, let's talk about multi-stability, because that is the main punchline of the paper, because that's what people really care about. Very few people care about enumerating all the states unless you are a big aficionado of the either stable marriage problem or maybe MacArthur model or something like this. So, but people care about multi-stability. So one take-home message here is that in this model, just like in the model I have shown you on a whiteboard, in order to have this multi-stability, regime shifts, hysteretic first-order transitions, species have to have different C to N ratios. So they have to consume carbon and nitrogen in different ratios. If everybody in the world consumes carbon and nitrogen in the same stoichiometry, the model becomes inherently boring and not worth discussing. Fortunately, it's not the case. As we know, there is some variation in stoichiometry of the species. Which species is feasible determined by supply fluxes. I use this in answering, I made this point in answering your question. And each state has a feasibility region, which means that if I am interested in this state, I can calculate, or maybe it's easier to go like this. So this is kind of a pictorial thing of what is happening. By the way, this is the only part of this. Actually, I think that most of this stuff is real data based on this little toy example of 2 carbons, 2 nitrogens, 6 stable states. So here is all possible transitions between states happening in this model. The red ones are hysteretic regime shifts. The blue ones are smooth transitions between states when one species disappears, the other one appears, right? And in order to find which one is actually true, for every state, you can, using linear algebra, determine this polygon in some multidimensional flux space where the state is feasible. Namely, the way you do it is you just start varying the abundances of microbes, individual microbes present in the state, all the way from 0 to, well, you can make them normalized to 1, then it will be between 0 and 1, making the sum of them equal to 1. And by swiping through this entire kind of polyhedron in the space of microns, you will get a polyhedron in the space of supply rates of nutrients. If 2 states border each other along the border, then this corresponds to a second order phase transition. You can cross from one state to another without any hysteresis, with just one species disappearing and one species appearing, maybe, or maybe just one species disappearing. But if you have a red line, then those two kind of regions in a multidimensional space overlap. And here is the bistability region. And as I said, there are plenty, even in this little toy models, there are 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 possible regime shifts. Now, okay, this is my favorite plot, not because I want you to understand it, but because already after I made it, I realized that it's almost perfect replica of the Kandinsky painting, right? So here, just for your entertainment, I have shown, or rather my students have shown, not just those 6 uninvadable states, but actually all 27 invadable and uninvadable states, including even the state which is dynamically unstable. And of course, here, instead of polyhedrons, we just drew some PCA ellipses just to give you an idea. So all of those transitions are happening in this space. And of course, this is a two-dimensional principal component, one principal component, one principal component, two. But in reality, it happens in the four-dimensional space of those flux supply rates. So it is a beast, right? But there are all those transitions happening in this phase space. Now, and just to blow your mind completely, this is what happens for 6 carbon, 6 nitrogen, and 36 species. There are 900 states. Stable marriage model tells me that there are 900 states. And here, just to spare you a little bit, I only show the regime shifts, the first order transition. So every line here means that there is a transition between the states. The size of the node is the volume, the phase volume of the state. So many of the states have small phase volumes. Other states have large phase volumes. And you have those distinct regions, which means that there are really some principal component analysis here. There are different kind of combinations of resources. In one combination of carbon and nitrogen resources, only this kind of cluster will be important. In another one, only this one will be important, and so on. So important, very important. I'm not saying that for a given combination of fluxes, 900 possible states will be possible. This is always a confusing point. Whenever I'm given this talk, people say, oh, if there's exponentially many states, it's a complete mess. No, if I'm just have my environment determined by my fluxes, here is what I get. Most of the times, if I just randomly sample my environment, I'm in this four-dimensional volume, in four-dimensional space of resource supply rates. Most of the times, actually, something like 95% of the times, I will have just one state. It will be monostable. There is only one possible stable state, uninvadable stable state. Sometimes they'll have 2. Sometimes they'll have 3. The largest number we found in this particular example of 6C, 6N, and 36 species was 5. Meaning that in this particular space, I could have a transition between 5 possible regime shifts between 5 possible states. And this is, by the way, how it looks like. If I now randomly sample my microbial abundances in each of those 5 states and then do a principal component analysis, I will see this is state number 1, state number 2, state number 3, state number 4, state number 5. You may ask me, what about those gray regions? It turned out that they are dynamically unstable states. And the property of my model, which I saw numerically, I'm absolutely rock solid that it's there. It even helped me to find numerical errors. Sometimes I thought that I found a violation of this rule, and then I looked at the code and I found that it actually was an error in the bug in the code, not the bug in the. So in my model for each, notice that I have 1, 2, 3, 4, 5 dynamically stable states and 1, 2, 3, 4 dynamically unstable states. So everywhere in this model, in every phase region, the number of dynamically unstable states is 1 less than the number of dynamically stable states. Why? Just kill me. One thing which all of us know is that if you have some one-dimensional system, and if this one-dimensional system goes to plus infinity, if there is some Lyapunov function, which, by the way, I don't know Lyapunov function for this model, but if there is one, which I strongly suspect there must be because I never see cycles or chaos or any funny business like this. And if this Lyapunov function goes to plus infinity on plus infinity and minus infinity, then the number of minima always 1 more than the number of maxima, right? This is calculus 101. But first of all, I don't know the Lyapunov functions. And second of all, this Lyapunov function would definitely not be one-dimensional. I am operating in a 4-dimensional space. Why the hell I have this rule? I have absolutely not a foggiest clue.
Why isn't it an
You mean not an exponential fit?
I mean, I can just fit it with a line.
Yeah, I can fit it with a line, then it will be exponential distribution. And Poisson, actually, I honestly don't know. It's probably, yeah, it is log, of course, yeah. So you see that this 95% have, 90% of phase volume have only 1 state, 5% have 2 states. I don't know, 0.5% have 2, and only you know, 3 points out of a million, which I sampled, have 5 states, right? So that's, but I'm really like for a while right now, I'm kind of slightly over this model. I moved on, but I was really racking my brain. What is the Lyapunov function and why? What is the effective one-dimensional? You can kind of see it here. If you want to transition from here to here, you have to go through this state. If you want to transition from here to here, you have to go through this state. But why it is like this, I have no clue. Okay, I have 5 minutes. It's just perfect timing. So let's talk about priority effects. We didn't talk about priority effects much in this. And this is, again, the snakes and ladders game, which I have shown you last time, yesterday. And we have a variant of the snakes and ladder game in this problem. And of course, depending on which fluxes I'm working with. So for instance, here I'm working in my trivial model in which I have 2 carbon sources, 2 nitrogen sources, 4 species, and each species can be either carbon limited, which I depict as red, or nitrogen limited, which I depicted as blue. I can start with an empty state, empty chemostat. Then, of course, I can add each one of the 4 species. And for this configuration of fluxes, those 2 species will become carbon limited, and those 2 species will become nitrogen limited. Now, depending on what I add next, I can switch between 2 alternative stable states. And priority effect is essentially when you are colonizing an ecosystem, the outcome of colonization depends on the order in which I add the species. Since I added the blue species first, then it only accepts another blue species. If I added the red species first, it only accepts the red species. And that's why once I add it up in this state, I cannot add any more species. And in order to make a transition to this state, I need to kick somebody out. Here is another, it's the same model, the same parameters, but in a different part of the phase space where I have 1 uninvadable state with 3 species. And now the transitions are, well, on one end more convoluted, but in the end, they lead to 1 final uninvadable state at the top of the pyramid, right? So everything here is more predictable. But some of the transitions are impossible, and also some of the transitions lead to rewiring. So for instance, here I had those 2 species. I added 1 species, which you can guess which one. By the way, a good question to you. When I make a transition from 22 to 26, which species did I add? Well, just think about which species is there which wasn't before. The answer is this one. So I added this species. It became nitrogen limited and kicked out this species, which was also nitrogen limited, which probably means that they were limited by the same nitrogen. Actually, they were limited by the same nitrogen. That's shown in the way they are positioned on this wheel of karma. Okay. The other thing, this is kind of a slide I like. So I have 36 species. And in this particular system, every species has a chance to shine in at least one of the states. So if I really, so some of the species, so again, the x-axis is average competitiveness rank of a species, meaning the rank on carbon plus the rank of nitrogen divided by 2. The best you can have is a species which is the best on its carbon and the best of its nitrogen. This guy is, of course, present almost everywhere. Now, actually, probably it's present everywhere. If I think about it, it should be present everywhere because it is really a Superman who will kick any other species from the system. But then there's this poor guy whose competitive ranks is 5.5, which means it's number 6 on carbon and number 5 on nitrogen. This is as low as you get, but still in 1 out of a million, 1 out of 1,000 of points in the phase space, it will be present. So the bottom line is that even if you completely suck, there is still room for you in this model. And the last slide, I think, I hope at least, is this, which is actually something which I can compare to the data. So you may say, okay, I can change the ratio stoichiometry of carbon and nitrogen in my supply pipes. I can make very carbon poor medium. The supply of carbon is 10 times lower than supply of nitrogen, or supply of nitrogen 10 times lower than the supply of carbon, or I can make it 1-to-1. By the way, 1-to-1 is just nominal in my model. I fully realize that in real ecosystem, C/N ratio is not 1-to-1. For simplicity here, I use it 1-to-1. If not, we need to rescale it to the average stoichiometry in, which in the ocean is well known. It's known as the Redfield ratio. But this is kind of the average stoichiometry of all species. And that gives me the highest diversity of all species, up to 11 out of 12 possible. But when I am in nitrogen limited or in carbon limited corner, the diversity falls. And that's very easy to understand. Just imagine that one of the resources is supply in abundance. Let's say in this case, it is carbon supplied in abundance and nitrogen is limited. That means that in this case, all the states I can predict without using any stable marriage. There will be a unique possible state here where every nitrogen will be occupied by the most competitive species for this nitrogen. Carbon will not be limited to any species. So there will be this hospital diagram will be just from every nitrogen species, there will be one arrow emanating to carbon. There will be no carbon limited species. The same is here, except that carbon is switched to nitrogen. And all of this crazy kind of interplay between arrows is really taking place in the middle. And this is something which, of course, ecologists knew for a while, that if you take an ecosystem and so we didn't discover it, it's actually a rather simple observation which have been made over and over. So this is another plot from Tillman, which he himself got from the data on some oldest experimental field station in the world, which is in London. By the way, the second oldest, which is only younger than this London field, is in Urbana, Illinois, and I'm looking at it from my window of my office. There is this particular plot of land which they have been studying since 1960 or something, and sorry, 1860. And this is 1860 is where this experiment starts too. So they studied it over 80 years, and they did 2 experiments. In a control experiment, they didn't fertilize those fields, and the diversity, which was a plant diversity at the time, stayed constant. But once you start fertilizing the field, which means that you take one of the nutrients, let's say nitrogen, maybe they fertilize with nitrogen and phosphorus too, but imagine for now that they dumped a lot of nitrogen, making nitrogen non-limiting to any species, and the diversity fell quite a bit, right? So that is exactly the same story as this story. Okay, now I think I have 1 minute, 30 seconds, which is perfect timing. First of all, I want to say, okay, I will not repeat these 3 points. You can read them yourself. I want to draw your attention to point number 4 and number 5. One is that what if you start to generalize this model to more than 2 essential nutrients, say carbon, nitrogen, phosphorus. I already told you last time that the 3-sex or 4-sex stable marriage is maybe unstable, and it will be unstable in this nasty way when you will have this recurrent breakups. Like first you will break up with this, they will break up, and then it will come back to it. So there will be those breakup cycles. This is related to this old result from Huisman and collaborators, which published in Nature 1999, that if you have more than 3 essential resources, you can have cycles. 4 and more, you can have chaos, okay? Now, the second thing is, you notice that all of my species are specialists, which is not a good thing to have because many species are not specialists, at least in carbon. So here is what I propose to do. I haven't done it yet, and it may not work with stable marriage approach, but it will still work. I propose to call it quantum marriage, and I plan to apply for funding from the Novo Nordisk Foundation, which are into quantum biology. Why it is a quantum marriage? It's because instead of having one spouse, you have a quantum superposition of several spouses, right? You plug in the MacArthur model on carbons, MacArthur model on nitrogen, and then you apply a mean function. And that will be the most realistic model I can have for carbon and nitrogen utilizing species at low resource concentration. So I'll keep you posted about my quantum marriage success. Okay, take-home messages. I told you 2 marriages, 2 stable marriages problem, right? One is one which was a theory, meaning that it was a theory in the search of application, and the application in microbial systems didn't quite work out because in chemostats is not happening like this was dioxic microbes. Tomorrow I will tell you how how it actually works with dioxic microbes in serial dilution systems, not in chemostats. But maybe it is, as Matthew suggested, maybe I thought about it myself before, that maybe it's the right model for prey switching, right? You know that some of the predators, they first eat rabbits, and then when they run out of rabbits, they switch to whatever, moles or something like this. So all you need to do is to implement in this prey switching behavior the fact that no more than one predator can eat a given prey at a time. And then it will be a model for prey switching. But in any case, I want to say that it was microbes was one side, carbon sources, substitutable carbon sources one other side. Stability was not an invasion, it was rather a microbe switching from one carbon source to another in a dioxic fashion. Now, the story I told you today is microbes are actually edges, microbes are marriages, and nutrients of two types, carbon and sizes, are, if you want, men and women, or hospitals and applicants. And now as I already told you, I already spoiled this joke, lovers are allowed as long as they are better than the spouse. And the invasion is, can a microbe like this invade? Because, for instance, if everything is limited by the blue guys, this microbe must outcompete this one if it invades. But can it invade or not is determined by a variant of a stable marriage model. And here, for specialists on two essential resources, is absolutely a rock solid rule. So if you ever encounter an ecosystem in your wanderings around where you will deal with a specialist on two essential resources, I will be the guy to talk. Thank you.